Hi, today we are going to talk about topoisomerase. Topoisomerase is an enzyme that is generally associated with dissolving supercoils. We are going to try to understand what are those and how exactly this enzyme resolves that and also the various types. Now we know that our DNA is a double helix. That means there are two strands of DNA which are termed as the strands, and we have the directions as well. So we say 5 prime and 3 prime because of the 5 prime has phosphate and 3 prime has the OH group, right? Now each turn has about 10 base pairs. Now how to imagine this? Imagine this as two ropes which are intertwined with each other. If you try to pull them, what will happen? The rope will overturn simply. But what if the end of the rope were fixed rather than being free to turn if they were fixed, and now you try to pull them apart? The distance between those coilings will decrease, and hence they will become supercoiled. Now, not all the DNA in the organisms or the cells are actually circular or fixed, but yes, we do have some circular DNA as in case of bacteria. But because of the huge size of DNA that is there in the chromosome, sometimes one end can be assumed to behave as if they were fixed. Now where this happens, we'll talk about that. Let's talk about the types. There are two types: positive and negative. So a positive supercoiling would increase the density and hence decrease the access to the enzymes, whereas a negative supercoil will decrease the density. It will unwind the DNA and hence increase the access to the enzymes. When the DNA is about to be replicated, what is replicated? Replicated means it is about to be copied. Now, a very huge amount of supercoiling can block the access for DNA replication enzymes. Let us look at how the DNA is copied. So we have two strands. As we can see, we have two strands of the DNA, and they are pulled by special enzymes called helicases. Now helicase, that means it is going to break the helix, right? Or in other words, it will going to pull apart the helix. So when the helix is pulled apart, our enzymes strand, so our DNA strands, the strands separate, and because they are separated, now the RNA primer can be laid down, and using the RNA primer, special enzyme DNA polymerase can start acting and add new nucleotides. But this situation is very same as me trying to pull the two ends of an intertwined rope. It will increase the tension. How to resolve this tension? For this we have topoisomerase, which cuts. So basically, if these are the two strands, topoisomerase can cut simply one strand. After this, this can simply flip over. Because of the cut, it can simply flip over and the tension is resolved. Such types of topoisomerases which cut only one strand, they are known as topoisomerase type 1. Now, that means there has to be a type 2, right? So the topoisomerase type 2 is a little bit more brilliant. What it does is it cuts both the strands, and then it allows it again to flip over, right? So topoisomerase 2, one of the famous topoisomerase 2 is also called by the name DNA gyrase. So DNA gyrase is another topoisomerase 2, an example that's found in E. coli, and it can also reduce tension by allowing the supercoiling to be resolved. Now you can imagine how important topoisomerases are, because without this, the supercoiling would become a big issue, and hence if the supercoiling keeps on continuing, then the DNA replication can come to stop. So DNA replication will come to stop if the supercoiling is not removed. That is why several drugs try to target topoisomerases, because we know that topoisomerase is a very important commodity when it comes to cancerous cells. Why? Because cancerous cells replicate a lot, need to replicate a lot. So because we want to preferentially target the cells which are replicating a lot, we have some drugs that attack these topoisomerases, and because of the attack, topoisomerases cannot work, and hence the replication cannot continue, because helicase would not be able to anymore open up the DNA and continue the replication. So I hope that helped. In today's class, we try to understand that DNA may have supercoils, and these supercoils can change access of the DNA to the enzymes. Topoisomerase control the supercoiling by introducing single or double standard breaks. And hence, it is very important in DNA replication, because without this, the helicase will not be able to proceed ahead, and hence the replication process will come to a halt. I hope this helped. Thanks.
Want to learn more? Check out the next video. Quick reminder to subscribe to GeeklyEDU.